Dissolution is the kinetic process where a solid substance enters into a solvent to form a homogeneous molecular dispersion. This transformation requires the physical detachment of molecules from the solid surface and their subsequent diffusion through a stagnant boundary layer into the bulk fluid. In the context of drug delivery, this phase transition is frequently the rate-limiting step for bioavailability, determining exactly how much of a compound reaches the systemic circulation. Observe how the surface area and solubility constants dictate the speed of this interaction as we begin our exploration of mass transfer kinetics. When dealing with poorly soluble compounds, the kinetic bottleneck for systemic entry is the dissolution phase, categorizing it as the absolute rate-limiting step for absorption. By quantifying this phase transition in a controlled environment, we can reliably forecast bioavailability, which represents the precise fraction of the active dose reaching the circulatory system. This essential correlation empowers research and development teams to refine chemical formulations while allowing quality control to ensure rigorous consistency across production batches. Consider the implications. If the rate of dissolution lags behind the rate of membrane permeation, the entire therapeutic outcome hinges entirely on this single physical transformation. Solubility represents a static thermodynamic limit, specifically the maximum mass of a solute, such as 50 milligrams per liter, that remains in solution after reaching absolute saturation. In sharp contrast, dissolution serves as a dynamic kinetic measure that tracks the percentage of drug entering the solvent at specific time intervals, establishing a quantifiable relationship between duration on the x-axis and concentration on the y-axis. Analyze the mathematical notation at the bottom, which confirms a direct correlation where an upward trend in solubility generally forces a corresponding increase in the dissolution rate. This interaction occurs because the saturation potential of the medium dictates the velocity of the phase shift, meaning that while solubility defines the ultimate capacity, dissolution characterizes the actual speed of the molecular transition. Compendial methods provide the standardized mechanical environments necessary to simulate the gastrointestinal transit of dosage forms with mathematical precision. Note how Apparatus 1 utilizes a mesh basket to contain the solid, while Apparatus 2 employs a rotating paddle. Each configuration generates a distinct hydrodynamic profile that directly alters the thickness of the diffusion boundary layer. Specialized configurations like the reciprocating cylinder and flow-through cell introduce variable agitation and continuous solvent exchange, allowing us to model drug release under conditions of changing pH or infinite sink volumes. Examine these 6 designs and consider which specific mechanical motion would best maintain a constant concentration gradient for a high-volume controlled release system. By selecting the appropriate apparatus, we fix the mechanical shear and mass transfer variables essential for validating the kinetic stability of any pharmaceutical product. Focus your attention on the architecture of USP Apparatus 1, where a stainless steel mesh basket serves as the primary housing for the dosage form during the dissolution test. The hemispherical design of the 1,000 milliliter vessel is a critical engineering feature that minimizes fluid turbulence, ensuring that the mass transfer kinetics remain consistent and reproducible throughout the experiment. We maintain the system at exactly 37 degrees Celsius to simulate biological thermal equilibrium while varying the rotational speed, typically 50 RPM for tablets and 25 RPM for suspensions, to precisely control the mechanical shear stress applied to the particles. Observe how this basket configuration effectively solves the buoyancy issue inherent in capsules by forcing the material to remain fully submerged within the solvent. Think about the hydrodynamic implications here. How does the rotational velocity of the shaft directly dictate the thickness of the stagnant boundary layer surrounding the mesh? The rotating basket is technically superior for dosage forms that exhibit buoyancy or require controlled containment, such as capsules and multiparticulates, because it prevents them from floating to the surface, where fluid agitation is inconsistent. A major scientific benefit of this configuration is the ability to execute a complete media change mid-experiment, which allows us to precisely simulate the physiological pH transition from gastric to intestinal environments without disturbing the solid-state orientation of the sample. Furthermore, the standardized geometry of Apparatus 1 facilitates seamless automation, ensuring that the mass transfer coefficients measured across thousands of routine quality control trials remain statistically consistent and reproducible. How would the release profile of a delayed release bead change if the mesh porosity was altered, thereby restricting the solvent flux into the basket? The fluid dynamics within Apparatus 1 reveal a critical flaw. A stagnant dead zone forms directly beneath the rotating basket, where the tangential velocity is insufficient to maintain uniform mixing. This lack of turbulence causes localized saturation, which directly interferes with the diffusion layer kinetics by preventing the rapid removal of solute molecules from the solid-liquid interface. Simultaneously, the fixed volume of the vessel poses a major challenge for maintaining sink conditions, which requires the bulk concentration to remain below 10 to 20% of the drug's saturation solubility. If the drug is poorly soluble, the concentration gradient, the mathematical difference between saturation and bulk concentration, shrinks so drastically that the dissolution rate artificially plateaus. Reflect on how these mechanical and thermodynamic constraints might lead to a false negative result when evaluating a potent but hydrophobic active ingredient. Transitioning from the basket, we encounter USP Apparatus 2, where a rotating shaft is equipped with a precisely shaped metallic blade known as the paddle. This configuration serves as the primary choice for tablets because it allows the dosage form to rest at the base of the hemispherical vessel, where it is subjected to the convective forces generated by the paddle's circular motion. The rotational speed of this blade dictates the fluid velocity, which in turn controls the thickness of the diffusion layer on the tablet surface and the rate at which the solid matrix erodes. Evaluate the physics of this setup. How might the shear stress exerted by the fluid change if the tablet is positioned off-center versus directly under the paddle's axis of rotation? Maintaining a standardized distance between the paddle and the vessel bottom is mandatory because this gap directly influences the local Reynolds number, ensuring that mass transfer kinetics remain reproducible across every trial. Apparatus 2 serves as a versatile workhorse for evaluating not just standard tablets, but also beads and enteric-coated systems that require a specific hydrodynamic environment to trigger drug release. Examine the technical specifications in the schematic. The paddle width and height are strictly standardized to ensure that the fluid velocity profiles generated are reproducible across different laboratory settings. The mechanical simplicity of this design allows for seamless integration into automated sampling systems, which maintains the integrity of the concentration gradient during the long-duration tests required for delayed release formulations. How would the disintegration kinetics shift if the dosage form moved from the central axis to the vessel's curved wall, where the tangential fluid velocity is significantly higher? By fixing the paddle geometry and rotational speed, we minimize variables in mass transfer, making this apparatus the gold standard for routine quality investigations. While Apparatus 2 is widely utilized, its design complicates multi-stage pH profiling because the absence of a containment basket makes the mechanical transfer of the dosage form between different media environments technically difficult. We also encounter thermodynamic limitations in the fixed 1,000 milliliter vessel volume. Specifically for poorly soluble drugs, the bulk concentration rapidly approaches saturation, violating the requirement for sink conditions and causing the concentration gradient to collapse according to the Noyes-Whitney principle. Physical irregularities like dosage form buoyancy or adhesion to the glass wall further disrupt the experiment by altering the local fluid velocity and the resulting thickness of the stagnant diffusion layer. If the sample floats into a lower velocity region near the surface, how significantly would the reduction in mechanical shear skew the calculated mass transfer coefficient? Consider these variables when selecting your testing parameters for hydrophobic formulations. Look closely at how we manage the physical positioning of the dosage form in Apparatus 2 when buoyancy or adhesion threatens the accuracy of our mass transfer measurements. When a capsule contains trapped air or a film-coated tablet becomes tacky, it may float to the surface air interface or stick to the glass wall, effectively removing it from the controlled hydrodynamic flow and causing an artificial reduction in the dissolution rate. To solve this, we employ inert sinkers, like the platinum wire coils or stainless steel springs shown here, to provide the necessary gravitational force to keep the solid fully submerged and centered at the base of the vessel. By utilizing these sinkers, we stabilize the local Reynolds number and prevent the reactive surface area from being obscured, ensuring the solvent flux remains mathematically consistent throughout the entire kinetic analysis. USP Apparatus 3 introduces a distinct mechanical dynamic where the dosage form is held within a glass cylinder that oscillates vertically between upper and lower wire screens. This reciprocating motion forces the solvent through the mesh, creating a high degree of turbulence that effectively strips away the stagnant diffusion layer surrounding the drug particles. Notice the reduced vessel volume of 200 to 250 milliliters. This smaller capacity makes the system exceptionally sensitive to the release kinetics of beads and controlled release matrices, where specific agitation rates are required. The unique architecture allows for the seamless transfer of the inner cylinder across a row of vessels containing different media, which precisely replicates the gastrointestinal transition from acidic to basic environments. How does the stroke frequency of this vertical oscillation influence the Reynolds number differently than the rotational velocity we observed in the paddle apparatus? Examine the mechanical sequence in the diagram, where the reciprocating cylinder transitions the sample between different media, a feature providing unmatched precision over simulated gastrointestinal pH shifts. This vertical movement generates intense fluid shear as the solvent is forced through the mesh screens, but we must meticulously account for the thermodynamic constraint of the maximum 250 milliliter volume. How does this restricted volume impact our ability to maintain sink conditions, specifically preventing the bulk concentration from approaching the saturation limit? If your formulation requires a large solvent reservoir to keep the concentration gradient high, the spatial limitations of Apparatus 3 could cause the dissolution rate to artificially plateau. The visual here displays the full instrumentation of USP Apparatus 3, where the reciprocating motion of the inner cylinders creates a distinct hydrodynamical profile compared to the rotational shear of the paddle or basket. Notice the alignment of the vessels. As the motor drives the vertical stroke, the fluid flux through the mesh screens at the cylinder ends directly impacts the Reynolds number, determining the efficiency of the mass transfer at the solid-liquid interface. This specific hardware configuration allows for a pre-programmed sequence where the inner cylinders move across rows, enabling us to model the entire gastrointestinal transit by transitioning the sample through a series of solvents with varying ionic strengths and pH levels. If we were to increase the dip rate from 20 to 30 strokes per minute, how would the resulting increase in mechanical agitation affect the thickness of the stagnant boundary layer and the subsequent rate of particle erosion? Consider the engineering precision required here. The timing of the dip and the clearance of the screens must be perfectly calibrated to ensure that the kinetic data remains reproducible across all parallel vessels. Apparatus 4 utilizes a flow-through cell, where the dissolution medium is pumped vertically upward through a chamber, creating a continuous flux that passes over the dosage form. This open-loop architecture is scientifically superior for low-solubility drugs because the constant introduction of fresh solvent prevents the bulk concentration from rising, thereby maintaining a maximal concentration gradient between the solid surface and the fluid. Observe the precise internal dimensions in the technical drawing, such as the 22.6 millimeter diameter. These fixed geometries, combined with the pump's volumetric flow rate, dictate the linear velocity and Reynolds number of the solvent as it interacts with microparticles or implants. If we were to transition from a laminar to a more turbulent flow by increasing the pump speed, how drastically would the reduction in the stagnant boundary layer thickness accelerate the mass transfer coefficient for a controlled release formulation? Apparatus 4 offers a sophisticated dual operational framework, allowing us to toggle between open and closed systems to manipulate the thermodynamic environment surrounding the dosage form. In the open configuration, the continuous introduction of fresh solvent ensures that the concentration gradient, the mathematical difference between the drug's solubility limit and its current bulk concentration, remains at a theoretical maximum, essentially eliminating the risk of saturation. Notice the integration of the media select valve. This engineering feature enables precise, instantaneous pH transitions to simulate the sequential movement through the gastric and intestinal tracts. However, the open-loop model requires massive quantities of media, presenting a logistical challenge in maintaining the exact chemical composition of the large fluid volumes needed to drive consistent mass transfer. Evaluate the hydrodynamic flow. How would a fluctuation in the piston pump stroke volume directly impact the thickness of the stagnant diffusion layer at the particle surface? Observe the vertical architecture of the flow-through cell, where the solvent is pumped from the base upward to establish a consistent fluid flux over the solid dosage form. By utilizing an open-loop configuration, we effectively decouple the dissolution rate from the bulk concentration, ensuring that the concentration gradient, the mathematical difference between the saturation solubility and the current fluid concentration, remains at a theoretical maximum throughout the entire assay. This constant renewal of the liquid phase ensures that we are measuring the intrinsic rate of molecular detachment without the inhibitory effects of local saturation that plague static vessel designs. Consider the kinetic impact of altering the volumetric flow rate. How would a surge in pump speed decrease the thickness of the stagnant diffusion layer and subsequently accelerate the overall mass transfer coefficient? Apparatus 5, known as the paddle over disc, modifies the standard paddle setup to evaluate transdermal systems by securing the patch to a stainless steel mesh disc at the bottom of a 900 milliliter vessel. We lower the operational temperature to precisely 32 degrees Celsius to mimic the cooler physiological environment of the human skin surface, rather than the core body temperature used for oral dosage forms. Notice the critical geometric constraint in the diagram. A fixed distance of 25 plus or minus 2 millimeters between the paddle blade and the disc assembly is maintained to ensure a uniform and reproducible fluid shear across the release surface. This configuration leverages existing hardware, but introduces a physical bottleneck, as the diameter of the disc assembly strictly dictates the maximum surface area of the patch that can be exposed to the solvent flux. How would the rate of drug diffusion from the polymer matrix be altered if the rotational velocity failed to overcome the stagnant layer formed within the narrow 3.3 mm disc housing? USP Apparatus 6 replaces the paddle of Apparatus 2 with a rotating cylinder, specifically engineered to evaluate the drug release kinetics of transdermal patches within a standard 900 milliliter vessel. To accurately simulate the physiological conditions of the human epidermis, we maintain the solvent temperature at precisely 32 degrees Celsius, which is significantly lower than the standard core body temperature used for oral dosage forms. The dosage form is adhered to the exterior surface of the stainless steel cylinder, meaning that as the shaft rotates, the tangential velocity at the cylinder's surface creates a uniform fluid shear that directly dictates the thickness of the stagnant diffusion layer. While this configuration successfully accommodates much larger patch surface areas than the disc assembly of Apparatus 5, the primary mechanical hurdle involves maintaining the structural integrity of the patch-to-metal bond under continuous rotational flux. How would an increase in the rotational frequency mathematically shift the Reynolds number at the patch interface, and how might that accelerate the depletion of the drug reservoir within the polymer matrix? Moving beyond standard dissolution vessels, the Franz diffusion cell provides a vertical bicompartmental architecture designed specifically to quantify the molecular flux across a biological or synthetic membrane. The drug moves from the donor compartment through a skin membrane, typically ex vivo rat tissue, into a receptor cell containing an isotonic phosphate buffer at physiological pH 7.4. This setup allows us to measure mass transfer driven by the concentration gradient, according to Fick's first law, while a water jacket maintains the precise 32 degrees Celsius thermal equilibrium required for transdermal analysis. If you were to collect a 200 microliter sample from the receptor compartment, what mathematical adjustments would you apply to the final flux calculation to account for the volume replacement and the resulting shift in the sink conditions? The kinetic velocity of dissolution is governed by 4 foundational variables: the intrinsic properties of the drug, the physical matrix of the dosage form, the chemical composition of the medium, and the mechanical dynamics of the testing apparatus. By reducing the drug's particle size, we drastically expand the total surface area exposed to the solvent, which directly accelerates the rate at which molecules transition from the solid state to the liquid phase. We must also account for the drug's chemical stability in the medium, because any degradation at the interface effectively alters the concentration gradient, which is the critical driving force for mass transfer. Furthermore, the dosage form type and the specific excipients used, such as binders or lubricants, will either facilitate or hinder the initial wetting and disintegration of the solid matrix. If you were tasked with optimizing a slow-release system, which of these 4 parameters would you modify first to ensure the drug reaches its systemic target at the precise therapeutic rate? The vessel's volumetric capacity serves as a critical thermodynamic regulator, scaling from just a few milliliters up to 4 liters to ensure we maintain the high concentration gradient required for sink conditions. This volume directly dictates the ratio of the mass of the drug to the solvent, preventing the bulk concentration from approaching the saturation limit, which would otherwise stall the dissolution velocity. Observe the two distinct geometric profiles, the hemispherical rounded bottom standardized by the USP and the flat bottom specified by the BP. These shapes are engineered to control the local Reynolds number at the base, with rounded bottoms specifically designed to eliminate stagnant dead zones where solute accumulation would artificially thicken the diffusion layer. Given these physical constraints, how would transitioning from a rounded to a flat bottom alter the mechanical shear stress experienced by a disintegrating tablet? The extent of agitation serves as a mechanical control that dictates the thickness of the stagnant boundary layer, where increasing the rotational speed effectively raises the mass transfer coefficient to accelerate molecular diffusion. We must meticulously regulate the temperature at 37 degrees Celsius for oral formulations to reflect core physiological conditions, while 32 degrees Celsius is the standard for transdermals to simulate the cooler surface of the skin. Furthermore, the nature of the medium involves selecting specific pH buffers that replicate the ionic environment of the gastrointestinal tract, which directly influences the drug's saturation solubility. By fixing these three critical parameters, we stabilize the variables in the Noyes-Whitney relationship, ensuring that the kinetic flux we measure accurately represents the release profile of the solid matrix itself. How would an unintended drop in vessel temperature mathematically impact the diffusion coefficient and the overall velocity of the mass transfer? To replicate the immense sink capacity of the gastrointestinal environment, the dissolution medium volume must be at least triple the volume required to reach the drug's saturation limit. Look at the math for a 5 mg dose with a solubility of 100 micrograms per milliliter. First, normalize your units by converting the 5 mg into 5,000 micrograms. By dividing this 5,000 microgram dose by the solubility of 100 micrograms per milliliter, you calculate a 50 milliliter saturation volume, which is the point where the solvent is essentially full. To achieve the necessary sink conditions, multiply this 50 milliliter threshold by 3, establishing 150 milliliters as your absolute minimum operational volume. How does this triple volume rule specifically prevent the bulk concentration from rising high enough to collapse the concentration gradient and stall the mass transfer process? Analyze the specific mathematical requirements for this 5 mg dose with a solubility profile of 10 micrograms per milliliter. First, convert the dose to 5,000 micrograms and divide by the solubility to determine that 500 milliliters of solvent is the exact threshold for saturation. Applying the triple volume safety margin for sink conditions requires a minimum of 1,500 milliliters, which ensures the bulk concentration remains low enough to maintain a robust concentration gradient. If this volume exceeds the capacity of your standard dissolution vessel, we must chemically enhance the medium by introducing co-solvents or surfactants to artificially boost the saturation solubility. Consider the thermodynamic shift here. By increasing the solubility constant through these additives, how does the resulting decrease in the required saturation volume allow us to maintain a constant mass transfer rate within a smaller physical footprint? To execute this experiment, we establish a controlled environment using 900 milliliters of either 0.1 molar HCl or pH 7.4 phosphate buffer at 37 degrees Celsius and a 50 RPM rotational speed to stabilize the mechanical shear. To track the kinetics accurately, we withdraw 5 milliliter aliquots at specified intervals and immediately replace them with an equal volume of fresh medium, a critical step to ensure the total volume remains constant for all mass transfer calculations. Once these samples are filtered and diluted tenfold, we measure the absorbance at the drug's lambda max to determine the concentration within the vessel at each time point. To solve for the cumulative drug release, you must multiply the measured concentration by the total 900 milliliter volume, and then account for the drug mass removed in previous samples. This allows you to determine the precise percentage of the dose released over the 60-minute duration. How would failing to perform that 5 milliliter volume replacement mathematically distort your calculated concentration gradient as the experiment progresses? Convert the UV absorbance reading into a sample concentration by multiplying it by K, the reciprocal of the calibration curve slope. Because you likely diluted the aliquot, you must multiply this value by the dilution factor, and then divide by 1,000 to shift your units from micrograms to milligrams per milliliter. Next, multiply this concentration by the total dissolution volume in the flask to determine the specific mass of drug that has entered the solution phase. To finalize your kinetic profile, divide this total amount dissolved by the initial dose and multiply by 100 to yield the precise cumulative percentage of drug release. Why is maintaining this rigorous sequence of unit conversions the only way to ensure your mass balance remains chemically accurate? Transform raw spectroscopic absorbance into a sample concentration by applying the constant K, which acts as the mathematical bridge between light intensity and molecular density. To reconstruct the true concentration within the large dissolution vessel, multiply that value by your dilution factor, and then divide by 1,000 to shift your units from micrograms to milligrams per milliliter. Next, multiply this concentration by the total dissolution volume to find the absolute mass in solution, then divide that mass by the initial dose and multiply by 100 to yield the cumulative percentage of release. Look at the column for actual amount dissolved. How would an error in your volume measurement at the 30-minute mark cascade through the rest of this kinetic percentage calculation? This kinetic profile illustrates the transition of a drug into the liquid phase over a 12-hour period, with the y-axis tracking the cumulative mass percentage and the x-axis measuring temporal progression. Notice the initial steep gradient during the first 4 hours, which represents a high rate of mass transfer, fueled by the maximal difference between the saturation solubility at the solid interface and the low bulk concentration. As the curve approaches the 100% threshold, the velocity of dissolution plateaus because the concentration gradient, the mathematical driver of diffusion, diminishes as the bulk solution nears its thermodynamic limit or the drug reservoir is exhausted. Examine the error bars at each data point. How does the variance observed at the 6-hour mark reflect the stochastic nature of particle disintegration compared to the more uniform initial release? By calculating the slope of this curve at any specific interval, we derive the instantaneous dissolution rate, which confirms the efficiency of the mass transfer coefficient under these specific hydrodynamic conditions. We define pharmaceutical quality through the parameter Q, which represents the precise percentage of the labeled active ingredient that must transition into solution within a strictly designated temporal window. This Q value acts as a legally binding kinetic signature found in the drug's official monograph, ensuring that every manufactured batch adheres to the same mass transfer rate. Observe the standard passing criteria for many immediate release products, where Q is established at 75% dissolution within 45 minutes to guarantee adequate systemic absorption. If a formulation's disintegration speed lags, how would that failure to reach the 75% threshold mathematically signal a critical breakdown in the consistency of the drug's delivery profile? Transitioning to official USP monographs, we see how the specific chemical properties of an active ingredient dictate the precise environmental parameters for mass transfer. For ammodia-queen HCl, we utilize 900 milliliters of water and a rotational velocity of 50 rpm to achieve a Q value where not less than 75% of the dose enters the liquid phase within 30 minutes. Contrast this with the sulfadoxine pyrimethamine combination, which requires a pH 6.8 buffer and a higher mechanical shear of 75 rpm to ensure each active ingredient hits a Q limit of 60% in that same timeframe. Why does this specific combination tablet necessitate a 25 rpm increase in paddle speed compared to the single-agent ammodia-queen to meet its kinetic threshold? By precisely tuning the medium's ionic strength and the rotational energy of the apparatus, we establish a standardized, legally binding kinetic signature that every batch must satisfy to ensure consistent bioavailability. This table outlines the statistical hurdles a batch must clear, starting with Stage 1, where all 6 initial units must individually exceed the threshold of Q plus 5%. If a single unit fails that high bar, meaning it dissolves at less than 80% when Q is 75%, we trigger Stage 2 by testing 6 additional units. For this total pool of 12 units to pass, the calculated mean must be at least Q, while a secondary safety check mandates that not one single unit falls below the floor of Q minus 15%. This two-tiered mathematical filter ensures that both the average mass transfer and the individual variability remain within strict therapeutic tolerances. Analyze this Stage 1 dataset where 6 individual tablets are evaluated to determine if the batch meets the initial kinetic threshold. For a product to pass at this phase, the USP mandates that every single unit must be greater than or equal to the Q value plus 5%. Looking at these results, the lowest dissolution recorded is 81%. If we assume a standard Q value of 75%, our passing benchmark is exactly 80%. Since every value in this table exceeds that 80% limit, the batch is officially accepted without moving to Stage 2 testing. How would the acceptance status shift if even one tablet registered at 79%, falling just 1 point below our Q plus 5 requirement? Observe the data for tablet 4, which dissolved at only 73% and triggered a mandatory transition to Stage 2 because it failed the Q plus 5 threshold of 80%. In this secondary phase, we incorporate 6 additional units and calculate the mean for the entire 12-tablet pool to ensure it meets or exceeds the target Q value of 75%. While our cumulative average of 86% successfully clears that hurdle, we must verify that no individual unit falls below the absolute floor of Q-15, which mathematically equals 60%. Since the lowest recorded value across both tables is 71%, the batch maintains the necessary kinetic consistency and is officially accepted. Analyze this specific dataset where the mass transfer rate of tablet 3 drops to 76%, violating the Stage 1 criterion of Q plus 5%, or 80%, and triggering an immediate progression to Stage 2 testing. By summing the dissolution percentages for all 12 units and dividing by 12, we calculate an arithmetic mean of approximately 73.7%, which fails to reach the mandatory 75% target established by the drug's official monograph. Even though the lowest individual value of 62% remains above the absolute failure floor of Q-15, which mathematically equals 60%, the failure of the cumulative average necessitates the complete rejection of this production batch to ensure therapeutic reliability. In this evaluation, tablet 3 clocks in at 76%, which is below the Stage 1 requirement of Q plus 5%, or 80%, necessitating an immediate transition to Stage 2 testing. We calculate the arithmetic mean for all 12 units to be approximately 79.1%, which initially seems promising as it clears the mandatory Q threshold of 75%. However, look closely at tablet 11, which records a dissolution of only 59%. Because the USP protocol strictly forbids any single unit from dropping below the Q-15% limit, which mathematically establishes a floor of 60%, this specific dosage form fails the kinetic uniformity check. Consequently, the entire batch must be rejected because that single outlier demonstrates an unacceptable breakdown in the mass transfer reliability. Comparative dissolution testing establishes a framework for evaluating the kinetic equivalence between different formulations or batches that share the same active pharmaceutical ingredient. To ensure the resulting data is scientifically robust, we must strictly standardize every mechanical and thermodynamic variable, including the apparatus type, solvent volume, rotational speed, and temperature. By eliminating these experimental discrepancies, we isolate the dosage form's structural properties as the sole factor influencing the mass transfer rate. What happens to the mathematical validity of your comparison if you fail to synchronize the sampling time points across all tested batches? Maintaining this precise uniformity is the only way to accurately map the dissolution profiles and confirm that two products will exhibit identical release velocities under physiological conditions. Observe how the reference and test formulations diverge significantly during the first 8 hours of the experiment, reflecting a distinct difference in their mass transfer coefficients from its solid matrix compared to the test sample. You must evaluate the error bars, the vertical whiskers at each data point, to determine if the variance between these two temporal release paths is statistically significant or merely an artifact of sampling variability. How would this shift in the dissolution rate constant affect the time required to reach peak systemic concentration, given that the cumulative release percentage over time is the primary driver of the absorption gradient? Observe this visual representation of comparative kinetics, where the y-axis tracks the cumulative mass percentage released into the solvent over a 12-hour duration. The high sampling frequency during the first 6 hours allows us to precisely map the initial burst and subsequent deceleration of the mass transfer rate for both the test and reference formulations. While the reference formulation shows a slightly higher initial release, notice how both curves ultimately converge at the 100% saturation limit by the final time point. If you were calculating the similarity factor between these two profiles, how would the consistent overlap of the standard deviation error bars influence your final mathematical assessment of their bioequivalence? We establish similarity through two specific regulatory gates, starting with the 85% rule. If both formulations release over 85% of their mass within 15 minutes, the dissolution is so rapid that the profiles are mathematically deemed identical without further analysis. When release rates are slower, we must calculate the similarity factor, known as F2, which is a logarithmic transformation of the sum of squared errors between the test and reference curves. Focus on the threshold where F2 is greater than or equal to 50, a mathematical limit that ensures the average difference between the two profiles does not exceed 10% across all sampling intervals. Why does reaching that 85% mark so quickly negate the need for this complex statistical comparison under current pharmaceutical guidelines? Examine the F2 similarity factor, a logarithmic equation designed to mathematically quantify the equivalence of two dissolution curves by measuring their cumulative distance. You must first calculate the squared difference between the reference and test percentages at each time point, sum these values, and divide by the total number of sampling intervals, denoted as n, to establish the mean squared error. This variance is then square-rooted and positioned within the denominator of the logarithmic expression, where the final multiplier of 50 scales the result to a maximum value of 100 for identical profiles. To maintain the statistical integrity of this comparison, ensure you utilize at least 3 distinct time points and exclude any data collected after both products have achieved 85% release. If your calculated F2 value falls below 50, what does that indicate about the average percentage difference between your test and reference formulations at any given moment? Apply the 85% rule to these two datasets to determine when a complex statistical comparison is actually mandatory. In example 1a, both the reference and test tablets surpass 85% release by the 10-minute mark, which automatically confirms similarity and renders the F2 calculation unnecessary. Conversely, in example 1b, the dissolution values at 15 minutes are only 72% and 78%, forcing us to calculate the similarity factor using the first 3 time points, where n equals 3. We stop our calculation at 20 minutes because once both formulations exceed 85% release, any further data points would artificially inflate the similarity score by focusing on the plateau rather than the active kinetic phase. If you were to include the 60-minute data in example 1b, how would that mathematically mask the real differences in the initial molecular flux between the two tablets? We must determine the precise number of sampling intervals, n, for our similarity calculations by identifying the exact moment both formulations cross the 85% dissolution threshold. In example 1c, observe that neither tablet reaches this threshold until the final 60-minute mark, requiring us to incorporate all 6 time points into the F2 equation to accurately quantify the variance across the entire profile. Contrast this with example 1d, where tablet A flashes into solution almost immediately, but tablet Y only hits the 85% mark at 20 minutes. Since we stop counting once both values exceed 85%, n is mathematically restricted to 3. Why would including the data points from 30 to 60 minutes in example 1d artificially mask the significant kinetic differences observed during the initial 20 minutes? By strictly terminating the calculation at the third interval, we ensure the F2 factor remains a sensitive measure of the active mass transfer phase, rather than the static plateau. USP Chapter 711 mandates an apparatus suitability test to ensure that the mechanical and hydrodynamic environment of your dissolution vessel is precisely calibrated before any experimental data is collected. For this performance verification, we utilize USP Prednisone tablets as a disintegrating type calibrator, which serves as a highly sensitive indicator of dissolved gases in the medium and irregularities in the agitation speed.
Simultaneously, we employ USP salicylic acid tablets as a non-disintegrating type. These function through a consistent surface erosion process, allowing us to detect even minor deviations in the fluid flow patterns or the alignment of the paddle and basket. If your measured dissolution rate for these standards falls outside the tight statistical range provided by the USP, how would that mechanical discrepancy undermine the kinetic profile of an unknown drug formulation? By verifying the hardware using these two distinct physical mechanisms, disintegration and erosion, we mathematically confirm that the mass transfer coefficient in our vessel is standard and reproducible. We validate the mechanical precision of our hardware using 10 mg USP Prednisone tablets, a disintegrating type calibrator specifically chosen because its dissolution kinetics are acutely sensitive to fluid shear and environmental disturbances. By strictly maintaining 500 milliliters of water at 37 degrees Celsius and a rotational speed of 50 RPM, we establish a controlled hydrodynamic environment to measure the cumulative drug release at exactly 30 minutes. To confirm that your apparatus is suitable for official testing, your results must fall within the strictly defined USP ranges of 47 to 82% for the basket and 37 to 70% for the paddle. If your data point lands at only 30% for apparatus 2, what specific mechanical misalignment or dissolved gas interference does that indicate about your system's integrity? This calibration ensures that every subsequent kinetic profile you generate is a true reflection of the formulation's properties, rather than an artifact of equipment variance.
We finalize the validation process using 300 mg USP salicylic acid tablets, which function as non-disintegrating calibrators to detect mechanical vibrations and misalignments through steady-state surface erosion. To execute this check, we increase the rotational velocity to a rigorous 100 RPM within 900 milliliters of phosphate buffer, while maintaining the standard thermal equilibrium of 37 degrees Celsius. Your success hinges on the percentage of mass released at exactly 30 minutes, which must mathematically align with the narrow windows of 23 to 30